The overall goal of the following experiment is to characterize circadian and sleep phenotypes in Drosophila by assaying daily rhythms of locomotor activity. This is achieved by generating transgenic flies, obtaining specific mutants, or setting up necessary crosses to prepare experimental animals. As a second step, glass activity tubes containing a food source need to be prepared, which will serve as the fly habitat during the experiment. Next, 1 to 5 day old flies are placed into activity tubes, which are loaded in DAM activity monitors in order to monitor locomotor activity rhythms using the DAM system. Results are obtained that show defects and variations in circadian and sleep parameters based on analysis of daily locomotor activity rhythms using software such as FastX and Insomniac.
This method can help answer key questions in the field of circadian biology, such as what proteins and regulatory mechanisms are involved in generating proper rhythms, and for example, how environmental factors such as light and temperature modulate behavior rhythms through specific molecular pathways.
The locomotor activity monitoring system involves numerous pieces of equipment, such as specialty monitoring devices, environmental incubators that have the capacity for diurnal light control, data collection devices, computers, and peripheral materials such as wiring. Choose a well-ventilated, temperature-controlled dark room to house the locomotor activity monitoring system to avoid excessive burden on the incubators and their possible failure to maintain temperature. Seal the room from external light sources. Inside the darkroom, it is not necessary to work completely in the dark, as the fruit fly's circadian system is not sensitive to infrared light. Therefore, in cases where we need to see in the darkroom, we simply use a standard flashlight that is covered with a red filter. Purchase an uninterrupted power supply emergency backup unit that has enough wattage capacity to power the components of the activity monitoring system in case of a surge, spike, or power failure in the building. Connect the UPS emergency backup unit to the emergency backup circuit of the building if available. If the system controlling the lighting in the incubator is not directly regulated by the incubator, then it is sufficient to plug the incubator into the emergency power without a UPS, as loss of power for a few seconds will not affect the chamber temperature. Next, set up a computer, PC or Macintosh, fully dedicated for data collection and/or for light control of the incubators. Manually arrange the telephone line network neatly around the shelving of the environmentally controlled incubators to allow ease of plugging and unplugging of activity monitors. Set up multiple telephone lines in a way such that they will converge into one main line and extend out of the incubator to connect to the computer. Connect the monitoring devices inside the incubators to the computer via a power supply interface unit, which serves to power the activity monitor via the telephone line. Finally, mask possible light sources from LEDs of electronic devices or an improperly sealed incubator door with duct tape or black cloth to ensure free-running rhythms are measured in the absence of unwanted light. It is crucial to assess these phenotypes using proper control animals that are reared in the same environmental conditions and of the same age. In addition, there is sexual dimorphism in circadian rhythmicity. The general practice is to use adult male flies for these assays that are reared at 25 degrees Celsius and between 1 to 5 days old, because egg-laying activity in female flies will affect true measurement of locomotor activity. When examining circadian and sleep-rest parameters of specific mutant flies of interest, it is prudent to outcross the mutant stock with the wild-type strain of the same genetic background, for example W1118 or YW. Since there is no crossing over in Drosophila males, it is better to perform the outcross by crossing mutant females with wild-type males. Seed both the wild-type control and mutant flies at the same time in standard Drosophila food for about 10 to 14 days before the locomotor activity rhythm experiment. Upon eclosion of the progeny, collect 1 to 5 day old male flies and set them aside to be used for the experiments. Activity tubes represent the fly habitat during the experiment. They are thin, 5mm glass tubes that contain food substance at one end and are plugged with yarn or a plastic plug at the other end. It is preferable to use activity tubes that are freshly made. They are generally prepared a few days to a week ahead of starting the experiment. Since glass activity tubes can be reused multiple times, the following preparation procedures describe using previously used and unclean activity tubes as the starting point. If you are using new activity tubes, simply skip to the autoclaving of the tubes as shown later. Remove the plugs from used activity tubes and put them into a large glass beaker. The tube should only fill up to half the beaker. Fill the beaker with tap water, making sure to submerge the tubes. Microwave the beaker filled with glass tubes until the water comes to a full rapid boil to melt the wax and agar food. Use caution as the water is hot. Remove the beaker from the microwave and stir the tubes with a spatula or plastic 10mm pipette to allow trapped wax to float to the top. Then repeat the microwaving step. Remove the beaker from the microwave and wait for it to cool down. Putting the beaker in the cold room will speed up the process. As the water cools down, the wax will collect on the surface of the water and gradually solidify. Remove the solidified wax by hand. This should get rid of most of the wax on the tubes. Transfer the activity tubes to a new beaker with fresh tap water and repeat the microwaving and wax removing process. Since most of the wax has been removed in the previous step, it is not necessary to wait for the wax to solidify. Simply pour the water out of the beaker and transfer the tubes into a new beaker. Use caution since the water is still hot. Repeat the microwaving process for the last time. Pour the water out of the beaker and wait for the activity tubes to cool down. Next, load the activity tubes vertically into 250ml or 500ml glass beakers. Make sure they are not too tightly packed. Sterilize them by using an autoclave with a dry cycle or simply dry the tubes in a drying oven. To prepare food to load into the activity tubes, make a solution of 5% sucrose and 2% bacto-agar in distilled or tap water. Autoclave the solution. The autoclaved food can be used immediately or stored at 4 degrees Celsius for an extended period of time. Ideally, the food should be around 65 degrees Celsius for filling activity tubes. Use a 10ml pipette to pipette the liquid food solution along the inside wall of the glass beaker, filling the tubes until they are 1/3 full from the bottom up. Swirl the beaker around gently to make sure that all the tubes are evenly filled with food solution. Wait for the food to solidify completely, either at room temperature or 4 degrees Celsius. Once condensation inside the glass tubes dissipates, the tubes can be removed from the beaker. To remove the activity tubes from the beaker, push the tubes towards the bottom of the beaker and twist the tubes at the same time so that the solidified food inside the tubes and the bottom of the beaker will separate. Take the tubes out of the beaker, preferably as a single bunch. Next, clean the tubes one by one with paper towels to remove excess food on the outer surface of the tubes. Set the tubes aside in a clean container. Take a general laboratory block heater without the tube holder and cover the heating well carefully with several layers of strong aluminium foil. Add paraffin pellets to the aluminium-lined heating well to melt. Hold the tubes at the non-food end and dip the food end into the melted wax. Dip the waxed portion into a glass beaker filled with cold water to speed up wax solidification. Repeat once. Dipping the waxed tubes into water will prevent the tubes from sticking together. The tubes can be used right away or stored in an airtight container at 4 degrees Celsius for use within a week. Prolonged storage will lead to excessive drying of the food. If the tubes are stored at 4 degrees Celsius, warm them up to ambient temperature by leaving them on the bench top prior to use. Prior to loading flies into activity tubes, Turn on the incubators that will be used to house the activity monitors. Adjust the temperature using the incubator controls and set the light-dark regime using the DAM system light controller or the incubator's own light control system according to the desired experimental design. Next, anesthetize the flies with carbon dioxide. Then, using a fine paintbrush, gently transfer a single fly into an activity tube. Grab the middle of a single piece of yarn that is around ½ an inch with fine forceps and insert the yarn into the non-food end of the activity tube to plug the opening and prevent the fly from escaping during the experiment, while at the same time allowing airflow into the tube. Keep the tube laid on its side until the fly awakens, to prevent the fly from getting stuck to the food. Insert the tubes into the activity monitors. With the newer, more compact model of the Trikinetics monitors, it is necessary to hold the tubes in place with rubber bands to ensure that the infrared beam passes the tube at the center position. Finally, put the activity monitors into the incubators and hook them up to the data collection system via the telephone wires. Check using the DAM system collection software that all the monitors are hooked up properly and that data is being collected from each of them. Now that the flies have been loaded into the activity tubes and the locomotor activity monitoring system is ready, the data can now be recorded. Flies are synchronized and entrained by exposing them to the desired light-dark, or LD, and temperature regime for 2 to 5 full days. The most commonly used entrainment condition is a light-dark cycle of 12 hours light, 12 hours dark at 25 degrees Celsius. For the study of circadian rhythms relative to this protocol, the time when the lights go on in the incubator is defined as Zeitgeber time 0, and all other times are relative to that value. Under standard 12 to 12 LD conditions, wild-type Drosophila melanogaster typically exhibit 2 bouts of activity, one centered around ZT0, termed morning peak, and another around ZT12, termed evening peak. Free-running locomotor activity rhythms are measured under constant dark and temperature conditions after the entraining period. The setting for the light cycle can be changed any time in the dark phase on the last day of LD, such that the subsequent day of the experiment represents the first day of DD. 7 days of DD data collection is sufficient to calculate the circadian period and amplitude or strength of rhythm of the flies. In general, a sample size of at least 16 flies is necessary to obtain reliable free-running periods for a particular genotype. At the conclusion of the experiment, raw binary data collected using the DAM system software is downloaded onto a portable data storage device, for example, a USB key. The raw binary data is processed using DAM FileScan 102X and summed into 15 and 30-minute bins when analyzing circadian parameters, or 1 to 5-minute bins when analyzing sleep-rest parameters. Currently, 5 contiguous minutes of inactivity is the standard definition of sleep-rest in Drosophila. Upon completion of this protocol, one can use the same dataset to compare the circadian and sleep parameters of the experimental and control animals. For circadian parameters, eduction graphs illustrating daily locomotor activities or average activities of flies over several days in LD or DD conditions can be generated using FastX. Drosophila melanogaster generally exhibit 2 bouts of activity, one centered around ZT0, termed morning peak, and another around ZT12, termed evening peak. These 2 bouts of activities are controlled by the endogenous clock and can be observed even in free-running DD conditions. Changes in the timing of these activity peaks can easily be observed in eduction graphs and may indicate a change in the properties of the endogenous clock. Another property that is indicative of proper clock function is the anticipatory increase in locomotor activity observed in LD cycles that occurs prior to the actual dark to light or light to dark transitions. This behavior is clearly observed in wild-type flies, but is absent in arrhythmic per-zero mutants. In this mutant, the observed morning and evening peaks in LD are purely startle responses due to abrupt changes in light-dark conditions. The clockless flies do not anticipate environmental changes, but merely react to them. Loss of behavioral rhythmicity is much more pronounced in DD and generally manifests into the total loss of morning or evening peaks of locomotor activity, as seen in per-zero flies. In addition to adduction graphs, locomotor activity data can be represented as a double plot actogram for each individual fly or fly genotype, where 2 days of data are plotted sequentially on each line, but the last day's profile begins the next line of 2 days' worth of activity. For example, LD1 and 2 are plotted on the first line of the actogram. The next line begins with a repeat of LD2 and is followed by LD3 and so on. Following this format, the locomotor activity data spanning the entire experiment is illustrated in the actogram. One advantage of actograms over adduction graphs is that a change in the period length of daily activity rhythms is easily observable. Besides generating adduction graphs and actograms, locomotor activity data from the DD condition can be submitted to FastX to calculate the period length using a number of different programs, including Cycle P. For analysis of sleep-rest parameters, which is defined as 5 contiguous minutes of inactivity, one can analyze data recorded from the locomotor activity assays and examine multiple sleep parameters using Insomniac, a MATLAB-based program. The percent of time that flies spend sleeping can be calculated at different time intervals. For example, percent sleep every hour or every 12 hours. Other more common sleep parameters that can be examined include mean rest bout length, which is a measure of how consolidated the sleep is and can illustrate the quality of sleep. Another is wake activity count, which is a measure of the activity rate when the flies are awake. This parameter helps to differentiate between flies that are truly affected in sleep-rest behaviors versus those that are sick or hyperactive. For example, flies that are simply sick may seem to sleep more because they are not as mobile. For these flies, their wake activity will be lower relative to control animals.
After watching this video, you should have a good understanding of how to characterize circadian and sleep phenotypes in Drosophila by assaying daily rhythms of locomotor activity.